Very good morning to all of you. Today's in the second session of this entire series. Our main agenda will be like how the proteins actually function. In this perspective, we'll be discussing how the proteins interact with each other and how the signals are passed. Like these signals govern the protein-protein interactions. These interactions govern the final functional aptitude aspect of the cell, like how the multiple proteins function. Because you understand that protein is not just an individual entity; it's a collection of 30,000 proteins in the human body. So how do they function? Every protein do not work individually, but it is working in synchronous combination with several other proteins. So they have to be like uh, working together to guide a single function. Anywhere the problem comes becomes a big problem for the overall pathway. Okay. With this big background, let us proceed further. The protein-protein interaction network is also termed as PPI or PAN, protein interaction network or protein-protein interaction network. Okay. But we understand that genes do not work independently. Okay. And the things keep on changing. Okay. Like if I take glucose, okay, different kind of enzymes will be produced in the human body. But the moment I take lactose, lactose will be first converted lactose present in milk. So if you take milk, lactose will be converted to glucose and galactose. Okay, so this glucose and galactose will further be metabolized into the same pathway as is done for glucose. Okay, but the initial one more step come in, and that lactase enzyme, after that glucose enzyme, the further take over. So the pathways keep on getting connected. Like that, proteins keep on interacting with each other all together to govern a single function. Okay. So all of us already know that DNA gives RNA to protein. This has been very basic, but now we understand that the proteins are governed by their final function. They also declare the phenotype, which is not just one protein, but a combination of multitude of multiple proteins. Okay. So biochemical conditions further define the phenotype. Okay. So protein structure and its topology defines the function. Okay. This is a basic gene regulatory network. Now, if you have taken it from a book, so if you understand it carefully, like if an input A is coming up here, these are any receptor proteins, transmembrane proteins present in a cell membrane. Okay, so there's a cascade of reactions will be coming up. Cascade of reactions means several reactions one after the other. A to B, B to C, C to D, like that. Okay, so signal A, which is a primary messenger, will now be transferred into a secondary messenger. This leads to like a inactive transcription factor A. It comes up. This becomes active transcription factor because of this. This input converts an inactive transcription factor A to active transcription factor A, one entity. On this side, another part, okay, leads to like input signal B. Okay, this converts like inactive transcription factor B to active transcription factor B. The inhibitor factor gets dissociated. Okay, so understand that active transcription factor A combines with B to form a protein-protein complex. This works together with the DNA regulatory elements. Here the target gene is there. Remember the last classes we have been discussing how the RNA polymerase starts the transcription. Okay, so there how many protein subunits are there? I hope you may be knowing. Alpha, alpha dash, beta, beta dash, omega. Five are there. Okay. So these subunits works together then form a holoenzyme, functionally active form of RNA polymerase. These five subunits combine together, and it all happens here. Any subunit not functional means RNA polymerase cannot function. Okay, so that you understand that it's such a complicated but beautiful process. Okay, so DNA phenotyping. Now understand the next concept before moving further. DNA phenotyping is a principle that if suppose I keep the DNA like this is a very basic diagram. Okay, I'm keeping the DNA like this. These indicate 1 to 22 indicate positions. Positions of different bases present on DNA. Okay, so 1, 2, 3, 4. If you check up in the population, what is the percentage of a specific base present in a population? Okay, that is shown there by minus log 10 bit, minus decimal logarithm. Okay, what is the probability of a certain specific base present in a population? This defines how many possible mutations are there at that particular base. Okay, so if more categories of bases are present on a single place, means there is highly variable in position. Okay, but somewhere it is different. So this is termed as genome-wide association study, which is used to for the SNP analysis. Okay, we'll be discussing later. We'll be discussing it later in the course. Okay, and all this can be predicted in much advance. Even before a baby is born, we can predict the entire SNPs, all the possible diseases coming up in the baby, right in the fetus itself. In the mother's womb itself, we can predict all this. Okay. So now understand that genes don't work independently. So I'm repeating here. Genes don't work independently. Okay, as per the conditions, they keep on switching between the protein partners and keep on working different fates, different protein signal transductions, different messengers. Okay, so different regulatory interaction mechanisms goes on. Okay, so this detailed mechanism is still under research. Okay. Here understand a beautiful term like if by a signal some protein expression gets increased. This is termed as upregulation. Because of certain messenger, certain protein expression got increased. Likewise, if because of some interaction of some protein, other protein expression got decreased. This is termed as downregulation. Okay, upregulation increase the expression. Uh, Down regulation means decreased expression. All this is caused because of one signal protein which has bound to this one. Okay. So now understand that this part I'm now again changing this gene information, molecular structure of protein, biochemical function, gives phenotype. From this initial part of basis of this entire today's lecture, I started this lecture. Now I'm taking this discussion further to molecular interactions. That all these make sense only when the complete network works in the way the cell wants. Okay. Anywhere a problem is there, the cell cannot function, and that will be fatal for the cell. The cell will die. Okay. See here. Okay, human necrosis factors is the TLR4 means toll-like receptors. Different proteins are there. How different proteins work in synchrony with each other. Okay, there's a possible part of possible regulatory mechanisms. Okay, this is highly connected network of various interactions, and that is a dependency. Until a protein function works properly, other one cannot do. Okay, So it acts as the rate limiting factor. Highly connected protein defines the key hubs. Just like in the computer systems, the hub is the main channel through which the entire network goes. Okay, just like a router is there in anybody's house. Okay, through which entire network traffic goes to the outside world and vice versa. That only controls. Okay. Likewise, some key protein defines the entire connected system of all the proteins. Okay, all the proteins are majorly governed by the regulation of that key protein, which is termed as the hub protein. Okay. Lower degree of connection, what is implies? Means certain protein may not be very essential in different conditions, but it may be essential in some other conditions. Okay, so see, the on-off switches we have discussed. Now understand this line. If certain proteins, they are always found interacting. I ask you the same question before I explain this line, which is underlined here. Some two proteins are always found interacting in different species. In different species, they are always found interacting. What it implies? This implies that neither of these protein can work independently, or they both require each other's presence to work. Any one of them is absent, it does not work. Means the way one protein is evolving in one species, in the in the same species, other protein also involves a similar way evolution. This is termed as co-evolution. Okay, understand the terms. These are very significant terms for because this course is advanced bioinformatics. Okay, this will help you in understanding several different research papers. The way one protein evolves, other protein also evolves same way. So if a mutation happens here, a compensatory mutation happens in the other protein partner, so that their interaction is always maintained. Okay, so this is termed as co-evolution. Okay, so see this part. Now, like if I want to, based on this alignment, I want to find out the evolutionary trees. This is just a small part to compensate that thing. I'll be coming back to the reason why I kept here. Different people keep phylogenetic tree. Okay, that certain things are found together as the leaves. We are calling them these two evolved from this parent, these two evolved from this parent, like that. But if I have this and I align them, I can always put a gap and keep it here. Okay, means they may be functioning together. So if one of them is performing some network, like network switch under some network switch that one protein is working, other one may also be working under the same condition. Okay, so whatever the conditions one is undergoing, the second protein partner may also undergo the similar constraints in the different species. Okay, so you can extrapolate that information. This is the entire principle of phylogeny. Okay, so if close relatives are found, there could be source of beneficial alleles that could be transferred to the other required species using genetic engineering. Okay, using DNA samples are now able to differentiate legal species from like illegal species of many organisms. Okay, so now understand what all, even if we take the sequences, normally what happens, we take many sequences, some of which are very close functionally, some of which may not be functionally closer. Okay, so their structures may be substantially different. So in that case, always remember what it can show. Even if you build Falcon tree for all this entire analysis, whatever we are discussing, patterns of descent, common ancestors, it can show 100%. But phenotypic similarity cannot be granted. Understand? Age of species, like how much old it is, okay, we cannot exactly say. Although the point accepted mutations and these kind of matrices can be used, okay, so how much to the probable like age of diversity which has happened, okay, you can calculate, okay. So understand that if close relatives are found, that should be the source of beneficial in alleles that should be transferred, okay, just to extrapolate the network information. See, the next part. The next application of all these sequences like motifs. Sequence motifs you can easily find out. Okay, from different sequences. So besides this network part, sequence motif is the next part. Sequence motif you can easily find out. What is motif? Motif means functionally conserved like a stretch of bases. Okay. So for DNA or protein, it may be termed as DNA motif, protein motif, sequence motif, structural motif, but the meaning is same. Motif means conserved part. So within a sequence, if some bases or some amino acids are conserved, which is functionally conserved in all the sequences of that same class, then we will call it as sequence motif. Structural motif means if some small structural repeat is present in all like functionally similar classes of all the same proteins, similar proteins, then it is structural motif. Okay, see, motif started here, gene starts. See, we can find. Okay, we normally find it up. It should be a consensus string, although it can have mutations. Means out of 6 bases, one amino acid, one position may be A or T can be there. Others are same, similar conserved bases, but at one position it may be A or T. Okay. So what are the complications coming up here? Okay. So whenever you are taking a new sequence, say suppose some student has found a new sequence from a bacteria, then he found that that bacterial sequence does not exist in NCBI database. Then what will do? Okay, he cannot find out the motif, he cannot find out the function. What is the thing? So, and here we don't know the motif also, no information at all. So in that case, how to start? Okay, so we do not know where it is located relative to the gene start. Motif can differ significantly from one gene to the next. Okay, then only the conserved motif concept turns up. How much conserved it is, at what base it is conserved, or where the variation turns up. Okay, so how do you distinguish from the random sequence segments and motifs? We know motifs are conserved. Okay, but if you don't know, anything can be there. Okay. Um, so in this part of the session, we'll be now extending our discussion onto the Human Genome Project. Like all these initial things led us to sequence the entire human genome and led us to a series of experiments down the track. Okay, so but before the Human Genome Project came up, taken up by the US, a series of several experiments went after the other. Okay, so we'll be initiating with that to conclude how it proceeded further. Okay, because all this is linked. So the entire part here is like how to crack down the code which is encrypted in the human genome. Okay, so that we can resolve many troubles like finding new drugs, or like finding how what is the genetic basis of different diseases, or how like different diseases are passed on in the entire like progeny, or the entire past, like from parents to children. Okay, all right. So the entire started from 1944, like where these three scientists, Avery, McLeod, and McCarthy, worked out. Okay, what they did was, I'll explain you in nutshell here. See this like diagram. What they did was like understand that rough strain was non-virulent, okay, and smooth strain was virulent, okay, having the that strain was virulent, okay. So when they infected the mice with the rough strain, okay, and they infected another mice with a smooth strain. Here the mouse died, mouse was killed, okay, and here was mouse was surviving, okay. So then what it implied means smooth strain is infectious, rough strain is non-infectious. Okay, that was the basic part. Okay, so now to understand that they mixed both of it and then again checked. Okay, then the mouse was like found dead. But when they were heating, they like heated up the smooth strain and then infected the mouse. The mouse was surviving. Means something was there in the smooth strain which was passing on to the mouse and then was killing the mouse. Okay, then was killing the mouse. Even when like heat killed this heat killed smooth strain plus rough strain, which was non-virulent, means something got transferred from smooth strain to rough strain, converting it into smooth strain. So now the question was, if something is getting transferred from smooth strain onto the rough strain to make it a infectious agent, what could be? So is it protein, DNA, what it is? Okay. So what they did was they removed all the proteins. They removed all the proteins from that heat killed smooth strain. Okay, on the smooth strain, they removed everything. So they found that it did not destroy its ability to transform R strain cells. Means mouse was again getting killed. Then what they did? Okay. Then what they did? Using a DNase, they destroyed the DNA. Okay. Thus the DNase is a DNA digesting enzyme. Okay, it breaks down. So now you understand that this entire conversion of heat killed smooth strain getting transformed into a deadly smooth strain. Okay, because something got like okay, something got transformed into like rough strain got transformed into the virulent strain. Okay, so because of the transforming agent. So but if you're now like using DNAs, if you're breaking down the DNA, nothing get transformed. So mouse is again surviving. So this indicates that DNA is the genetic material. Okay. So because of this entire series of experiments, they concluded that the DNA is the genetic material. Okay. Further, after to confirm it, few years down the line, in 1952, Hershey and Chase came up. What they did was they took bacteriophages. As you know, bacteriophage is a virus which infects. Okay. So when they are found that like to explain it further, they considered that okay, see here, it was having heat killed same part, heat killed estrogen plus this lipids, sugars, so it has protein, RNA. So they are using proteinase to remove the proteins, RNAs to remove RNA, DNA to remove DNA. Okay, so they are just finding, confirming it whether it's a DNA, RNA, or protein. They are there to confirm that it is DNA, but they are again confirming it. Is it DNA, RNA, or protein? Understand? To further validate it using more sophisticated enzymes, rather than using the heat gain. Okay, earlier experiments used just heat. So here they are using proteinase, DNase, RNase separately and then working it out. Okay. So when if you are adding proteinase here, no protein, H cells appear. Okay. Smooth stain is still appearing when it's virulent. Here RNase is there. See, R cells, H cells appear. Means when you are adding R cells and something is coming up from the top, okay, then it is transforming R into S. Because if R cell is there, it is absorbing something which is coming up here, which is present in the mixture. Even after using RNase or proteinase, the transforming agent is still not like removed or broken down. The this non-virulent R strain is still getting converted to virulent S strain. So what could be the reason? Okay. So what could be the reason? To understand it properly here, see, then they used DNase here. Then they found that okay, if they are adding R cells again, now this time if you see, no S cells appear. S cells did not appear at all. So after every MacLeod and McCarthy experiment, Hershey and Chase further confirmed it. And here they did it entirely using the radioactive materials P32 and S35. The P32, you understand, it is present in DNA. S35 is present only in proteins. So why they have chosen this only? Because these elements are specifically present in DNA and protein, like no other thing. Other elements are present there, are present in these molecules. Okay, so this finally confirmed that DNA is the genetic material. Okay, understood? Okay. So now I'm entirely clustering this series of experiments rather than going in detail of each of it. I'm now clustering it in this way. See, Gregor John Mendel gave the Mendelian genetics, of course. He initially turned up that something is there. Okay, DNA is there. Then every MacLeod and McCarthy experiment came up. Okay, Hershey and Chase came up. Then the Watson and Crick model came up. DNA is double stranded. Then this codons, bases, everything, the entire concept came up. The 1977 further series of experiments, the see, sequencing and other things. Here the PCR came up. This finally led to Human Genome Project. Okay, we'll start Human Genome Project in 1990. Okay, they started. US started. Now they have taken 13 years to complete the first part. Complete the first part here. They took 13 years. See, so much complicated it was. Okay. So this led to the human genome, discovery of human genome finally came up in 2003. If you understand the series of experiments now, whatever I just discussed, 1953 the Watson and Crick gave the model using X-ray diffraction. Uh, I hope you remember the X-ray diffraction model which they used using the Bragg's law. Okay, like using the lambda part, the wavelength, they calculated the angles and other things, how it got diffracted, everything. They placed using the top. Okay, but now after 1953 it came up to 2003 when the first sequence came up. Okay, so now understand that it contains 3 billion nucleotides. But the magic is 3 billion nucleotides when they are mapping into different genes. It was found that only 30,000 genes now. Now we know it. That nearly 30,000 genes exist in the human genome. In 3 billion bases, only 30,000 genes. Okay, so how much it is? It's only 1.4%. See, only 1.4% of the genome is encoding for functional protein product. Okay, so what is the function of 98.6%? We don't know. But what the researcher says, that is have to something to do with the functional part, like how to regulate the things, under what conditions to regulate what, or how much to control. If some protein expression is going on, how much it should be expressed. All those parts is taken care. And these chromosomes have the 23 pairs are there. One is sex chromosome XY or XX in females, XY in males, and 22 pairs, normal pairs. Okay, these are termed as autosomes. See what it led to. What it led to is like researchers decide the goals. 2003, the first map of first blueprint of human genome came up by the US Department. Okay, Department of Energy. But like it led to the series of experiments. Okay, we need to map different genes. What is the sequence of different genes? Like how to map different genome of different human entire population. Okay, so determine the sequence of 3 billion base pairs. Fastest way. Initially, the sequencing was taking too long time. Okay, the Sanger sequencing initial methods which got developed, but then it now came very fast, very cheap sequencing. Okay, because if we need to sequence the entire human genome of many entire population, okay, then we need to have the cheap for fastest way. And if the data is coming up means we need the sophisticated databases so that we can always compare our sequence with the known sequences and find out, okay, this gene is here, this gene is here, this is the function of gene, this is the function of this gene. Okay, the annotation, this is the term of annotation concept, okay, which I just talked in the last lecture. Always remember my series of lectures so that you understand the concept. If the data is coming up, databases are done, well improved, well updated. We need tools also for the data analysis. Okay, because of which the BLAST and other tools came up in 1994. Remember, 1990 this was like it was got started. The moment some data came up, the BLAST was coming up, basic local alignment search tool. Okay, transfer related technologies to the private sector. The reason is when it's government sector, it was taking too much money, too much time. So private sector, it could be done much faster. So if the private sector comes in means some ethical issue comes up, like ethical, legal, and social. If I want to sequence your genome, you will definitely think why somebody should sequence my genome, why somebody should store my genome in their databases. What is the wrong thing they can do with my genome? Will they do some harm to me? Yes, basic common question which is there. And these were the milestones. You see? 1990 it got started. 2000 we completed 90% of the genome. And that means at least the entire genome. We are having 3 to 4 redundant copies at every place so that we can be confirmed that this is the entire sequence of this part. This is the confirmed sequence. Until the duplication is there, until the redundancy is there, like we don't know, is it right or wrong. So we need to repeat the same experiment multiple times. Okay, 2003, the first human genome project sequencing got completed. See, 29,000 bacterial artificial chromosome, which can store a large information, were taken up for storing 3.2 billion base pair of the human, nearly 3 billion base pair, 4.3 billion base pairs. Understand that first of all, don't get confused in the numbers. 3 billion base pair, 3.2, 4.3. Understand that human genome is very big. See here. So when I'm using the building the bacterial artificial chromosome library, I'm chopping down the DNA just like scissors at different parts, random places, and then I'm putting up all that together, all those big fragments into the clones. I need to clone it. Okay, like I'm building the bacterial artificial chromosome, I'm putting it there, just like the plasmids. I'm inserting the like my gene of interest in the plasmids. Same way back clones were made. Then I see, got it them sequence. Okay, then I got them sequence here. See, then once I'm getting the like shotgun clones, all these parts properly, I can get the entire sequence of all these fragments. And now I need to link these fragments. Understand? Here, like understand it properly. The genomic DNA I'm taking, I'm cutting it at several different places, and then I'm like all these parts I'm putting in bacterial artificial chromosome clones, back clones I'm developing, building a library for it, and then I'm sequencing all of it, and then once those sequences are built, I'm now overlapping all of them in the computer, so as to make up okay 3 to 4 like sequences are there for every place. Okay, redundancy, and then I'm making the final part. Okay, this is done. Within this, but with the length of these segments, it will be substantially large because multiple fragments may be duplicated. So this is that number. Okay, containing large amount of repetitive DNA. Are you understanding? Okay, so this was done. See, I got everything here. So now after doing the assembly here, after assembling the entire genome, like I got the final sequence here that this is the entire sequence. Okay. So this led to the next part of the human genome project phase. We'll be discussing what are the basic concepts which they have now taken to link the part or how it came up. Okay. It's a primitive part which is taken up. Okay. Genomic DNA here, you got the different fragments. You overlap them here. Yes? So this is termed as aligned contiguous sequences, one after the other. Okay? See? Like suppose if I ask you ABC, then BCD, then DEF, EFG, like that. I give you 4 fragments. ABC, BCD, uh, CDE, then DEF. Okay, or an EFG, like that. So when I'm combining them here, two bases are always I'm putting repeat. The last 2 bases I'm repeating in the next one. So what do you think? So it will be very common sense, very clear that the 2 bases are repeats. So I'll ABC, then BCD fragment is there. I'll insert D here, ABCD. Then CD fragment is there. I'll insert CD. I'll add E here. Likewise, I'll append F here. Likewise, I'll append G here. So like that, I can build the final sequence. Okay. This part was termed as aligned contiguous sequences were termed as contigs. Okay. See, I got in merged data here. I merged the entire sequence. I got the final sequence contig. Small pieces combined together. See, when it reaches the bigger length, I was calling them as scaffold. Small pieces, contigs, contiguous strands, contiguous sequences. Then when it's big length now, I'm calling it as scaffold. See, so now like big fragments, scaffold, the one scaffold, two, like that. So now I'm linking the scaffolds also. Now I'm combining the scaffold. So contigs, small pieces lead to scaffold, then lead to mapped scaffold. Now I'm putting them together. Okay. And this STS was sequence tagged sites. I was knowing the place, that where exactly it is. So the reason is somewhere if it's ABC or CBA, I can know that this position, this length is STS. So it should be present here only. Just like we use milestones, milestone on any like road, we are using milestone. Likewise, this concept was there. Okay. So short sequence that can be easily detected, and whole genome location can be properly mapped. Okay. So contigs for only 200 to 800 base pair, only this size. So when we went to scaffold, it went on to substantially large sequence segment, 2 to 40 kb, large amount. So now you understand that 3 billion base pairs means much more larger, much more larger. So then scaffolds are also combined. Okay, see, this is the entire part in a nutshell. So it's the entire game of overlapping the parts. See, 27 million reads, 16 libraries, finally linking into the complete big genome. See, this is the same concept which I was telling. How do you link it? So this end-to-end alignment is there. See, if its middle overlap is in the middle, it is not okay. But if it is end-to-end alignment, you can always link it further. Such things were discarded. Such things were taken forward. See, and all this was done by computer with human interventions, so as to properly give the segments. Only overlaps were needed so as to like properly be confirmed. And so what is now the application? Now the application of all this is in the medical science. Genes and human diseases we can map. What is the genetic basis of cancer? Like why in certain genes are only having the cancer? Or what happens when the cancer turns up? Drug target identification. Okay, we can easily find out like what is the possible target for this like disease, or what is more conserved in this disease, so unique in this disease, so that I'll build my drugs only against that. Pharmacogenetics, like some diseases work against something, some diseases work against something else, so I can link genetics with the drugs. Okay, why in certain things it's coming up. See, no benefit, no toxicity, nothing happens. But the disease, like medicine, also don't help. Benefit, no toxicity at all. The medicine is not toxic to them. So this is what is expected. Here the benefit is there, toxicity is also there. Means side effects. No benefit plus toxicity. This diverse spread of disease, same medicine, but four different effects are going on. And all this is done because of their genetics involvement, the sequence of DNA involved there. Okay, the gene therapy. This is the last part. Like here, like if I am knowing the gene, I can always treat it to correct SNPs. Part of this. Now we are reaching to the last part of this. Last part of this entire session. Here we will be discussing the important concepts before moving to the next sessions. That is, what is the structure of protein, like different macromolecules, giving rise to protein data bank (PDB), macromolecular data bank database (MMDB). Okay, and what is the need of Ramachandran plot? Okay, understand that proteins or macromolecular structures are more important for us rather than the sequences. Reason is, if you know the structure, you can exactly find out the function, exactly find out like their interaction with different ligands or inhibitors. If the interaction is going on, how the interaction is changed with different ligands, which amino acids are involved, what are the bonds formed, everything. Every single thing regarding the structure function relationship we can extract only from the structure. Okay. All right. So students, now we are coming out to the next part, last part of this. Here we will be discussing what all databases are there. Okay. So here understand that the major functions of databases are store, display the viewers are also there in the database tool like home page. They should be able to collect the data from different authors. If data is there, we can be able to communicate. Okay, so there's a hyperlinks. You can always search or execute the scripts. Okay, the APIs, application programming interfaces are there, or the exchange. Okay, there's like always exchange of material. If one database wants some information from the other database, there should be always connectivity between the databases. Now understand that I'm categorizing into like structure, sequence, and others. Structure, you know, like protein database, SCOP, and CATH. These are two definitions through which PDB databases are again categorized. I will be showing it here. The SCOP and CATH, structure classification of proteins, like class, architecture, topology, homology. These are two different definitions, okay, through which you can classify all the protein structures in different classes, okay, functional classes. Then if the sequence databases are there for the proteins, it is SwissProt. You understand that the SwissProt database is a manually annotated database, unlike like a Uniprot or the Universal Protein Knowledge Base Uniprot, or like the DNA database of the DNA protein database like of NCBA. There people are manually just dumping the sequences. But whatever the function written there for any sequence may not be the same, as I told you in the last lecture. So SwissProt is the manually annotated server. Protein information resource. Here, what is the class, what is the family, what is the function, what is the different ligands which are interacting, all different species like functional information along with their cross reference to different other websites are also present in this server. Okay, then Tremble. As we discussed, Tremble is translated European Molecular Biology Laboratory. Okay, so here not only the basic sequences, they translated protein sequences are existing. Okay, so you can always find out the pro site information. Okay, the pro site gives information regarding the same as PAR. Okay, it is giving the information about proteins, but just the display analytics are different. The PFAM is protein family database. Now we are clubbing the proteins into families and superfamilies based on functions. Okay, I'll be showing here what is the definition of families, superfamilies. All that is coming from SCOP and CATH. Okay, so I'll be showing here. Please wait here. Okay, so nucleic acids. When I'm coming to nucleic acids, understand there is a basic gen bank which is there in NCBI. Then DDBJ, DNA Data Bank of Japan, European Molecular Biology Laboratory, MBL. Okay, these are there. Here you can just type the sequence name, DNA kinase, you'll get the DNA of different nucleotides of different species. You can classify them, you can always segregate them, you can compare it with different other sequences. Then some other databases are also there. And in others, many lists are there. It's a substantial big list depending on different class. What is the function? But I'm showing here three. So Rayleigh base is like how the proteins are involved in the different different ligands and other things. Okay. Then rebase is restriction enzyme database. You can always compare. Okay, like different restriction enzymes, what all are there, what is the function, what is the entire part coming up. Or GPCR, like G protein coupled receptor. This GPCR is involved in the body in several different places. Like those big proteins involved in several different important functions. So because of that, a separate databases is there. Okay. Yes. Now understand the SCOP and CATH story. Understand. I'm putting it here. See, class, fold, superfamily, and family. Understand it. Whatever the way I'm showing here, see this only to understand better. When I'm putting class, okay. So in the class, SCOP says that there are four classes: alpha helix, beta sheet, alpha slash beta means like beta helix is normally parallel beta sheet, okay, towards the same orientation of the like since on the same side, parallel beta sheet, outside covered by the alpha helices, okay. And other one is the alpha plus beta, where both are distinctly arranged and the beta sheets are normally anti-parallel when you are combining them together into fold. So all these fold and other things, family are coming up when you are considering multiple similar sequences. Based on that, the definition is evolved. So because now our objective is to cluster different protein sequences or structures so that the meaningful information can be derived. Understand? So fold means like there's a similar arrangement of super secondary structures, alpha slash beta, Rossmann fold, different serpentine 7 helix bundle arranged together. That is arranged same way in different proteins. So I'll say this 7 helix bundle is present as a fold. This is a important fold which is existing in nature but may be present in different functions. Okay, so based on similar secondary structure arrangement with same topology. Topology is a mathematical term, same arrangement. Superfamily, so based on that protein, when I'm considering multiple different species, multiple different sequences together, I'm calling it a superfamily. This leads to family. And now coming back to CATH. CATH is very specific. Here it's only considering three classes: alpha, beta and alpha beta. Then it's saying architecture. Based on the structures only it is doing. Architecture based on the orientation. Topology based on connectivity. See, homology. All the homology are based on the implying the significant part. Significant structural similarity and evolutionary data. It's considering the homology here. But I really like SCOP because of more definitions there. You can put more properly. Okay. You can refer to this Malik et al. 2020 paper. See here. As we all know, these are the levels of protein structure. Why I'm putting here is just because many of you may not know, remember, may not know it properly. So this is understand that the primary means primary sequence, basic peptide sequence, basic protein sequence. When it's forming alpha helix or beta sheet, okay, it's termed as secondary. When those secondary structures combine together, it forms tertiary. When those tertiary structures combine together, it forms quaternary. But how it is coming up here primary, it's only because of assembly of amino acids. It is because of folding of proteins, folding of amino acids, how the things are uniting together. Okay, then its second tertiary structure is related to packing, how the packing of different like secondary structures is going on to give rise to a specific tertiary structure, and how those tertiary structures are interacting together to form a compact unit that is termed as quaternary structure. Okay, in between these there is a super secondary structure. You remember Rossmann fold and other things. Okay, so that fold means nothing but alpha, beta, alpha, or any different parts. Okay, so that specific arrangement of secondary structure, it's termed as super secondary structure. So because tertiary structure is the combination of multiple different secondary structures. Okay, but in between that only few are arranged in a very specific manner, and that is always retained. It is termed as super secondary structure. Okay, this is because of protein domain. Reason is that will always function same way. Okay, so I can call it as protein domain 1, domain 2, domain 3. So likewise I can categorize like a form a diagram of my entire protein as domain 1, domain 2, domain 3, like with the specific arrangement of super secondary structures plus the same arrangement with the same function always. Okay, like that the structures are categorized. Okay, even the tertiary structure is also categorized as within one or two domains, three domains, like that, and that is also termed as subunit. Because protein has got its own subunit. Subunit 1, subunit 2, like that. Okay. But when its subunit concept changes when it comes to a big complex, protein-protein interaction complex. There one subunit of a complex means one tertiary structure or one protein. Okay. Remember the terms are same but the meanings are different. Okay, see? So all these parts is written here also. You can see it here. Okay. So as you can see it now, like these are the structure databases. The PDB like stores the three-dimensional information. Okay, so three-dimensional means X, Y, Z. Okay, here we store the data obtained by X-ray crystallography, NMR or computer modeling. Now, can somebody tell me which one is more accurate? What is more accurate? Here, the X-ray crystallography is highest accurate. It can reach up to zero 0.9 Armstrong resolution. Okay, resolution means if two dots are present together, how much clearly you can separately distinguish them as two different dots. Okay, that's not just one dot, that's a two dots. So if I'm saying like two dots are present at a distance of like 10 centimeter, you can easily see that these are two dots. But when I'm bringing the distance between them closer to 1 centimeter, then also you can see them as two dots. But if I bring the distance between them as 1 Armstrong, you will 100% see that as one dot. But still, they are separated by 1 Armstrong distance. So X-ray crystallography can still demarcate a line of 1 Armstrong between them. That yes, these are two dots, not just one dot. Okay. NMR has a resolution of nearly 1.8 to 2.5, and computer modeling. Okay. We are modeling the sequences based on these known structures. We'll be discussing it later. Okay. MMDB, macromolecular databases. So over 28,000 now 3D protein structures is there. See? Now the protein structure itself is like crossing 1.5 lakh, 2 lakh nearly. So we are reaching a substantial explosion. Some years down the line, like nothing was there earlier. Like it was taking too much time because X-ray crystallography was taking nearly 3 to 4 crore rupees, Indian rupees, plus 3 to 5 years. Okay, because the protein crystal needs to be taken up, then X-ray and structure need to be analyzed, then complete this XYZ coordinates. As I told you, no, three-dimensional atomic coordinates. XYZ coordinates need to be found out. Everything need to be done. But now it is much more better. It's much more better, much faster. See, and this is the home page. Home page of this PDB database. Okay. This entire like protein structure database is maintained by like RCSB, Royal Consortium of Structural Biology, a group between multiple scientists of all together. Okay, Indians are also there. So they are like what are the structures they are there? They are depositing the structure. So it takes some time for the PDB, and then they review the structure submitted by different authors, and then they put it there. See, I've shown it corona part here so that you can easily remember here. See? Alright, so in the last part of this session, we'll be discussing the Ramachandran plot. Here, what is the concept is this G.N. Ramachandran was 1963 working as an Indian scientist in Indian Institute of Science, Bangalore. He wanted to study like if you put up some amino acids as a like he studied with alanine, X alanine, where X can be amino acid, any amino acid. So if between those amino acids there will be one plane here, okay, and one plane on this side. So how the phi and psi angle okay, will vary depending on the x. If the x changes, how the phi and psi will change, just to pack them energetically in the lowest possible structure. Okay, that was the entire part. So to study that, what he did was, so he was saying that the energetically favorable region means something where the x is being placed. Okay, so that will take up the energetically favorable conformation depending on the phi and psi. So what all phi and psi will be taken up by the particular x. So starting with 20 natural amino acids, like it formed the multiple tripeptides and then worked out for different tripeptides what all like conformations or what all how many possible position in the maps that can take. Okay, to be showing the energetically visualize the energetically aroused regions for the backbone diagonal angles for the phi against psi. Okay, if I plot a map against phi and against psi for amino acids in the protein structure, it will show that. So this will be some regions which are allowed, some regions not are not allowed. Okay. So now, but if you are seeing the current maps of the Ramachandran plot, we are seeing that some regions are allowed, some regions are generously allowed, where we change some restrictions and generously allowed, then forbidden map, forbidden part. Okay. So, and this can be used for the structure validation. For any unknown structure, if you are considering the Ramachandran plot, and that Ramachandran plot says only 40% of the structure is fitting in Ramachandran, means the amino acids are not properly packed, means the structure is completely wrong. Whatever the structure you have done, calculated for these amino acids, okay, for this considered sequence, that is wrong, because it is not the way these amino acids should be properly placed in the energetically favorable conformation. All right. So see, so all right. So see, this is an example of a Ramachandran map. See, there are specific diagonal angles taken up by different like secondary structure elements, anti-parallel beta sheet. See, N to 3 and C to N next see in the opposite side. The parallel beta sheets is the same position. Okay, and then they see the right beta sheets right twisted, see, coming up here, left handed alpha helix coming up here, right handed alpha helix coming up here. See, they're all taking different parts. Okay, so this entire part like minus 180 to 180 degree for the psi, minus 180 degree to 180 degree for the phi. The phi psi map can be taken up, and depending on that, we can calculate whether the experimentally determined protein structure or like modeled protein structure is right or wrong. Okay. So here I have reached the end of this session too. Thank you and good luck to all of you.